Well, thank you all for coming. Welcome to Wave Club. My name is Josh Jacobs, and I help organize Wave Club along with Bar Dermantrout, Uma Mohan, and Erfan Zabeh, as you can see here in our nice little screenshot intro slide here. Our presenter today is someone I've known for a while, who I'm really excited to have here. His name is Mark Kramer. Mark Kramer is at Boston University. He's going to give a talk today titled Observations and Models of Traveling Waves During Human Seizures. Um, and Mark is a professor of math and statistics and computational neuroscience at Boston University. His background includes a PhD in applied physics from UC Berkeley. And he does a wide range of work, often focused on epilepsy that includes mathematical and computational neuroscience, neuropopulation dynamics and brain rhythms. And in addition to the mathematical work, applied work analyzing brain signals and healthy cognition and neurological disorders, along with some collaborators, people that I happen to know, including Kathy Siobhan. I think he's going to talk about her work too. But really trying to tie these mathematical models into neurological diseases, especially epilepsy. And he's co-authored as well statistical analyses of neural data. So that's really cool. So before we dive in and let Mark take over the stage here, I just wanted to remind everybody a little bit about how Wave Club works. This is a monthly seminar talking about traveling waves and across the brain in general, humans, animals, simple systems, complex systems, behaviors. Our Wave Club talks are generally recorded and put on YouTube. So if you are in, for example, Australia and you don't want to wake up at 3 o'clock in the morning to watch our talk, you can watch the recording of it. If you go to youtube.com/@waveclubseminar, you can find them pretty easily. I believe there are 14 videos online right now, which I'm pretty proud of, thanks to the work of Uma and Erfan putting those together. And also, a good way to learn about our upcoming Wave Clubs is to follow us on Blue Sky. If you just Google Wave Club Seminar, you will find us and you can follow us and learn about things. We also have an email list that you can learn about as well to get notified of upcoming speakers. We have the presentations about once a month. Usually we have a 40-minute presentation with some time for questions at the end. The best is to type your questions into the chat, and then we can make the Q&A be a little bit interactive as well. And if you are interested, so that's the best thing, but also little clarification questions are welcome in the middle. Mark is game for that. And then finally, we are also always interested in new speakers for Wave Club in the future. If you're interested in speaking at Wave Club, please email me or Erfan or Uma or Bard. So that's how that works. I'm going to stop sharing my screen here and turn the screen over to Bard. I mean, to Mark, excuse me. Thanks so much for coming, everybody.
Yep, thank you. Let me get my share going here. Okay, and what do you see?
Perfect. Looks perfect.
This one
dive in. All right.
Okay, good. Okay, thank you. Thank you for inviting me and for accommodating my schedule. The last time I was invited, I had a fever, which would have been probably an interesting talk, but hopefully this one will be more informative. Today I will discuss observations and models of traveling waves during seizures. This is highly interdisciplinary work, involves mathematicians and statisticians, clinical collaborators, and engineers. Really, the work couldn't be done without all of them. I'm just presenting what we've all done together. And if you do have questions, do feel free to ask. Okay, let's start with an outline. So I'll begin with some motivation and data why we're interested in this problem. We'll then show some examples of traveling waves during human seizures. I'll discuss two competing theories to describe the origin of these traveling waves, and then we'll propose a resolution for the controversy surrounding these theories. Okay, so some motivation. Epilepsy is a disease that affects 65 million people worldwide. It manifests as seizures, which are multi-scale phenomena, all the way down to channelopathies, and then convulsions that involve the entire central nervous system. There are many important open questions in epilepsy. For example, how do seizures start and stop? What is the optimal treatment to prevent seizures or cure epilepsy? And the question we will focus on here today for this audience, how does seizure activity propagate across the brain in humans? So let's start by thinking about some patient data, again, to motivate us. We'll consider here a patient with pharmacoresistant epilepsy, which means that drug therapies have failed. So this patient has chosen to undergo an alternative therapy, and that's resective surgery, where the goal is to find the area of the brain that's causing the seizure and surgically remove it. As part of this process, there's lots of imaging that's done. And this patient also had invasive electrode recordings. So these are electrodes that are placed directly on the brain surface to observe the voltage activity. I'm going to show you some of the data. I'll warn you, some of the builds of these slides will be somewhat gruesome. I'll tell you before they come on. So here's the first one. This is a craniotomy from a human subject. The skull has been removed. Can you see my mouse? Look for a nod here. Yes, I see a nod.
Yeah, we see it.
See it. Okay. The skull has been removed. There's an 8x8 grid of electrodes that's placed directly on the brain surface. There's also a strip of electrodes that tucks under the brain, and then 2 depth electrodes that pierce the brain. The spacing between these grid electrodes is 1 centimeter. And if we look at the voltage activity recorded from a subset of those electrodes, 10 electrodes as a function of time, this is about 100 seconds of data, and I'll tell you something about this data. A seizure occurs sometime during this 100 seconds. And you may not be an epileptologist or a clinician, but you can probably guess where the seizure starts and stops. So it manifests in this particular way in the voltage activity recorded directly from the brain surface. And if you're looking at these data trying to decide where the seizure starts and stops, that's the same analysis that's done in the clinic. So the standard clinical analysis to drive the surgical resection, what area of the brain should be cut out, is visual inspection. So an epileptologist who's looked at thousands or tens of thousands of hours of data can stare at these traces and decide or help localize where the seizure starts and define the surgical target. That's not our goal today. Instead, we're going to focus on a different question. How do we characterize the spatiotemporal dynamics of these data? Okay. So what I showed you initially was standard clinical macroelectrode recording. What's interesting about these data is we also have microelectrode recordings. To illustrate that, so here is a cartoon representation of the patient's brain. You can see the 8x8 grid of electrodes and other strip and depth electrodes. There's also this red star, and this red star corresponds to a microelectrode array. It's 4 millimeters by 4 millimeters on a side, so it's very tiny. It's a 10 by 10 array of electrodes. And each of those electrodes records the voltage activity from a small neural population. So in this patient, there's simultaneous recordings from macroelectrodes and microelectrodes. And I'll show you some pictures of these implantations. And this is the last set of gruesome slides. Here's, again, an implantation of a microelectrode array. You can see it here. There's the microelectrode array, sorry. There's the microelectrode array. And then here is the microelectrode array with the macroelectrode array. So again, I'll put an arrow to the microelectrode array. You can see it here along with the macroelectrodes. So all of these electrodes record voltage activity simultaneously from this patient. And let's look at an example of some of these voltage traces. So first, the macroelectrode recordings. These are the standard clinical recordings. Here are 10 example electrodes. There's a 10-second scale bar shown here. And the clinician, the epileptologist, has determined where the seizure starts. It starts somewhere else, so not at these electrodes. It starts somewhere else. And then it terminates on the right side of this screen, over here on the right. Sorry, let me move my little window here. Okay. And simultaneous with these macroelectrode recordings, there's also microelectrode recordings. So here are 10 example microelectrode recordings, again, recorded simultaneously. And if we look at these data, maybe at the highest level, the thing that we will notice is that the dynamics look qualitatively similar. So we see some kind of change in activity about midway through this trace, an increase in amplitude of this activity at both spatial scales. And then the activity maybe becomes more rhythmic, and that rhythm, maybe we see the spacing between these large bursts of activity gets wider and wider until the seizure terminates. So qualitatively, the dynamics look similar across these spatial scales. Okay, so these are voltage traces. So now we're looking at the data at different spatial locations as a function of time. Let's also look at the data as it evolves in space. So to do that, I'm going to show you a movie. I will start by orienting you to what's happening in this movie. So let's start by looking at the top left. This is a 3-dimensional reconstruction of the patient's brain. You can see the 8x8 grid of electrodes here. some of these electrodes have labels, A, B, C, D, all the way up to F, and those correspond to the traces shown at the bottom. Seizure onset is this red line, and the movie frame we're looking at above corresponds to this green line. So warm colors here indicate higher voltages, blue colors indicate lower voltages. So again, this green line is this frame, and then plus or minus 250 milliseconds around that frame is shown here on the right. So to zoom in on those 6 voltage traces. You might notice this little red X that corresponds to the location of the microelectrode array, and that's shown on the right. So here's the 10 by 10 microelectrode array. It's tilted to match its orientation on the brain surface. Again, some of those electrodes are labeled A through F. Those correspond to the traces shown here at the bottom. This frame corresponds to the green line, and here's a zoom in around that green line, plus or minus 250 milliseconds. So there's the movie. There's going to be a lot happening here. I'll suggest you look at the microelectrode array to start. I'll play the movie, and if it doesn't come through, I'll just pause it at particular points and we can look at it. So let me know if it's not working. But I'll play the movie, and you'll see a wave of activity that moves across the microelectrode array, followed by some more complicated dynamics. And now I'm watching this trace on the bottom right. I'm watching for this wave to come across. I can see it's coming, and then I'm going to watch up here, and I'll see a wave of activity move across the microelectrode array, followed by more complicated activity. And here's another one. I can see it coming. It's going to move across the microelectrode array, followed by something more complicated. And if we do the same thing on the macroelectrode array, we can kind of watch for a wave to come. And maybe if you squint your eyes and look at this enough, you can convince yourself you do see a wave of activity moving across the macroelectrode array, although it's harder to resolve. The spatial resolution is not as good. Can you see, is the movie playing?
Sure.
Yeah, you can see it. Okay, it's getting through on Zoom. Okay, so I'll just let this play in the background as we're kind of 2/3 of the way through the seizure, approaching seizure termination. And maybe what you'll be able to convince yourself is that there's very rich spatiotemporal dynamics. It looks like there are waves of activity. I see one here, especially in the microelectrode array that we can see propagating across this microelectrode array, and there's also more complicated patterns of activity. This is not necessarily a new observation. So for over a decade, these types of recordings have been made in humans, and this observation of traveling waves has been known. And in particular, these waves across the microelectrode array, I think, are the easiest thing for us to see by eye. And what I like about this is you don't need any data analysis. We can look at these data and we can see these waves moving across the microelectrode array. Okay, I'll keep going. We can come back to this movie and watch it more if you'd like. So how do we go beyond visual inspection and try to characterize these traveling waves? There are many ways to do this. We're going to choose one. We're going to compute the coherence. And here's an example of the coherence between one microelectrode and one macroelectrode. Vertical axis is frequency, 0 to 30 hertz, and the horizontal axis is time. This is one example seizure. Seizure onsets at time 0. Coherence is shown in color. Warm colors indicate high coherence. And what we find in this example is that the coherence manifests towards the end of the seizure. So this is approaching seizure termination. It has some interesting changes. We won't go into that here, but the thing to notice is that the coherence does increase during the seizure, especially in the second half of this seizure. And what's nice about this coherence measure is we can then use it to estimate the phase delay between different electrodes, and then use that to estimate the group delay, the temporal delay between electrodes at some position on the brain surface at one of the macroelectrodes relative to the microelectrode array. So what do I mean by that? We're going to take all of the electrodes on our microelectrode array, and we're going to ask what's the group delay, what's the time delay for a wave between the A macroelectrode and this microelectrode array. And we'll do that for all of the macroelectrodes. So we're figuring out the time delay of these waves to hit the microelectrode array and macroelectrode array. And I'll show you a picture of that on the next slide. I'll call this a delay map. So it's kind of like we're centering these delays at the microelectrode array, and we want to look at them across the macroelectrode array. And let's look at some of these delay maps, see what we find. So here's an example delay map early in the seizure. So what's plotted here is delay in milliseconds on the vertical axis, and then spatial location on these two horizontal axes. Each of these dots corresponds to a macroelectrode. So if you can imagine taking this plot and rotating it and looking at it top down, you would see the macroelectrode array. So each of these dots corresponds to a macroelectrode. And if we look at this picture, we see some delays that are positive, some that are negative, again, all relative to the microelectrode array, but it doesn't look like there's much spatial organization. We can characterize that by trying to fit a plane to these delays over space. And if we do that, in this case, we have no evidence of linear spatial organization of these delays. And again, this is early in the seizure. Now let's go to a later part of the seizure consistent with the movie I showed you earlier. And if we look at the delay map later in seizure, we see that these delays become much more spatially organized. So the delays are positive on one side of the macroelectrode array relative to the microelectrode array and negative on the other side. And now if we try to fit a plane to this, we do find a significant fit. So the slope's non-zero. We could see that there is some constant, or there's some spatial distribution of these delays that's organized. And it's consistent with the idea of a traveling wave that starts at one side of the macroelectrode array and then moves across it. So we have some evidence of this shown here. What's nice about fitting these linear planes to these delay maps is that we can estimate interesting quantities, like where does this wave come from, its source direction, and also its speed, which is the reciprocal of the slope. I won't go into the speed right now. Let's look at the source direction. Where are these waves coming from? So let's look. So here's an example of source directions during one seizure from one patient. And we could estimate these source directions. So this is the angle in radians as a function of time, where we've normalized time here. So seizure onset occurs at time 0. We normalize time so that 1 is seizure termination. And we can average these results across different patients and seizures that have seizures of different durations. So we've normalized time in that way here. If we look before the seizure starts, we do see some waves that are moving across the surface, but they're not very organized. They tend to come from different directions. It's only later in the seizure that we see a consistent source direction of these traveling waves appear. So approaching termination, these waves are manifesting and they're coming from the same direction. We can characterize that by computing the direction consistency, which is asking how consistent are the locations of these gray dots. So that consistency is low before the seizure starts. It increases at seizure onset, and then it becomes even higher in the middle and late parts of the seizure. Again, this is one example, one seizure. If we look at a population of patients and seizures, we see the same effect, is that this direction consistency increases approaching seizure termination. So these waves are appearing, and they seem to be coming from about the same direction over and over again as we approach seizure termination. Okay, one observation. Now, with those observations, we're seeing these traveling waves, their directions tend to be consistent. We can ask, what mechanisms do we think are producing these traveling waves? To do that, we'll consider here a mean field model. We choose this model to match the spatial scale of our data. We're not looking at individual neurons, so we won't build a model at the scale of individual neurons. Instead, we observe the population activity, so let's create a model at that scale. Here, we're going to use a model that was originally developed over a decade ago, or 2 decades ago, by the Stein-Rosses. The model consists of 2 populations, an excitatory population and an inhibitory population. They'll interact through synapses and will also allow, when these populations are active, they'll send potassium into the extracellular space, which can then feed back and increase the excitability of these populations, so depolarize them. This is one node in our network. We'll add another node to this network so we can connect them. We'll have long-distance excitatory synaptic connections. We'll allow gap junctions between inhibitory cells, again, following the structure of this model. And we'll allow this potassium to diffuse in space. So it can move from node to node across space. So here are 2 nodes in our network. We'll build a 100 by 100 grid of nodes of these mean field models with the goal of modeling the cortical surface, this 2-dimensional surface. And when we simulate this model, we'll consider 2 things. We'll consider the activity of the excitatory population as representing a microelectrode recording. And then we'll average some of those. We'll average some neighbors and have that represent the average activity you would see at a macroelectrode. So that's a setup for the model. Let's simulate it and see what we find. And we'll first consider the temporal dynamics that we see at one point in space. So choose one point on this grid, this 100 by 100 grid. And let's look at the temporal activity. So here is a simulation. We have a macroelectrode average activity shown on top. Again, this is the activity of the excitatory populations. And then a microelectrode activity shown at the bottom. So a 10-second scale bar shown here. To induce a seizure in this model, we'll take one node in our network and we'll depolarize it. We'll increase its excitability. So after we do that, this one node will send a wave of activity across the surface. Kind of see the sentinel spike that goes across the surface. We then have these complicated dynamics that evolve at both spatial scales until approaching the end of the seizure, we see this much more regular dynamics. How do we terminate the seizure? We're going to turn off that one node that we activated, so we'll hyperpolarize it, and we should go back to some baseline state that's consistent with what we saw before the seizure. So here are some temporal dynamics at these two spatial scales from our model. Let's see what it looks like now in space. So we'll look at some snapshots of a movie of this simulation. So let me orient you to that. So here's a spatial map at one moment of time of our 100 by 100 grid. Hopefully this comes through. The grid is 30 centimeters on a side, again, 100 by 100 nodes. Active regions will be indicated by black, and then the white regions will indicate low activity or inactive regions. So we'll see a bunch of these spatial maps. Here are some snapshots of these dynamics as the simulation evolves in time. So the simulation starts at the top left corner and then moves from left to right, top to bottom. Initially, not much is happening. There's a low level of activity in our network, and then we start the seizure. We activate one node in this network. We depolarize an excitatory population at one node, so that's done here. This node sends out a wave of activity, and it induces these spots that we see. So we have regions that are active, shown in black, and then inactive, shown in white. And you see they're kind of interspersed. These active regions are sending potassium into the extracellular space, and that's diffusing and increasing the overall excitability of this entire surface. So as they're firing, they're sending more and more potassium to the extracellular space. It's spreading around, and we see that as time evolves, the excitability is increasing. We get these local waves of activity, and you can see them here. These local waves, they look like these little moons that propagate out and collide, propagate out and collide. Again, this is all sending more and more potassium into the extracellular space, becoming more and more excitable until ultimately we see traveling waves that are emitted from our initial active source. So this region is more excitable than anywhere else. It's now able to drive waves that spread over this entire surface. This continues until we inactivate that source. So we shut it off. There's one last wave that propagates, and then we return to this background level of activity. So I'll just mention that this model is able to produce many of the things that we see in vivo. There's one dominant source direction that emerged later in the seizure. It has things like the right speed of those waves. I won't go into the details. If you're interested, you can look at this paper. Instead, I'll tell you something else, some initial conclusions. And I'll call these initial conclusions the Boston hypothesis. So the initial hypothesis from our group was that there's a spatially fixed source that emerges during the seizures, especially late in the seizure, and is driving these traveling waves in a single consistent direction. So conceptually, we might think of this as we have some radio tower that's sending out these waves over and over again late in the seizure, and we're seeing them hit our microelectrode array. So there's one hypothesis for the source of these fast traveling waves during the seizure. I'm now going to propose a different hypothesis, but before, let me mention that this result is based on 3 patients. So we looked at 3 patients. It's very difficult to get these data. So 3 patients doing this analysis, and we find this observation of these fixed source driving these fast traveling waves. Okay, that's just one theory. Here is an alternative theory. And this alternative theory is based on the idea of an ictal wavefront. So I'll read it out here, and then we'll look at some data and try to unpack this definition. So an ictal wavefront is a slowly advancing, narrow band of continuous or tonic multi-unit firing that leads the seizing territory. Slowly advancing, so there's a slowly moving thing. It's narrow, less than 2 millimeters, and it's represented by lots of action potentials, multi-unit firing. Here's an example of this. So this is from Kathy Chavon's group, Columbia. They are also looking at microelectrode arrays implanted in human subjects. So here's a microelectrode array, and in this case, they've denoted 4 of these electrodes shown here in red. So the multi-unit activity, which you could think of as the average action potentials of a large population of neurons summed up, they're showing this multi-unit activity here as a function of time. Note the scale bar here is 1 second. And if you look at this picture and you look at these triangles, you'll note that this tonic firing appears first at electrode 4, and then there's this increase at 8, 29, 48. So we have this movement of this multi-unit activity across this microelectrode array. And it's very slow. It takes about 4 or 5 seconds to move across a very, very small distance. Remember, these microelectrode arrays are 4 millimeters on a side. So this thing is moving very slowly across the microelectrode array. So that's a slowly advancing narrow band of continuous multi-unit firing. Narrow because we could see it at one microelectrode. We move over a little bit, again, a tiny interval in space, and we don't see it there. Okay. So the ictal wavefront. Here's an example of it from a human subject. Here's another cartoon illustration of it. So we imagine there's a microelectrode array, it's implanted in a human subject, and then this orange region represents the ictal wavefront that's shown here. As the seizure evolves, this ictal wavefront slowly expands over the cortical surface. It crosses the microelectrode array, and that's when we see this increase in multi-unit firing that we saw in the previous slide. It then continues to evolve. The microelectrode array is now stuck in this post-recruitment region inside of the boundary of this ictal wavefront. And ultimately, the ictal wavefront dissolves and the seizure ends. And what's interesting about this theory is that traveling waves propagate in a very specific way relative to this ictal wavefront. And let's look at a cartoon to see how these things evolve, and then we'll look at some data. So to summarize, these traveling waves, these fast traveling waves are going to propagate away from the ictal wavefront. So here's a cartoon of that. We're early in the seizure. We'll have an ictal wavefront shown here as this black dashed line. It started at this star, and now it's slowly evolving over the cortical surface. We'll also have a microelectrode array shown here. Now, this ictal wavefront is going to be sending off fast traveling waves in two directions. It's going to send these fast traveling waves ahead of it in the pre-recruitment area and also behind it into the post-recruitment area. So the ictal wavefront is going to be sending out these fast waves in two directions as it slowly expands. And these waves will be sent from all corners of this ictal wavefront. So it keeps sending out these fast traveling waves, including across our microelectrode array. So these fast waves moving in this direction across the microelectrode array. Time evolves. The ictal wavefront slowly expands. It passes over the microelectrode array, and now it has a much larger boundary. So this ictal wavefront is still sending off fast traveling waves in two directions. It's sending them into the post-recruitment region, into the interior, and also ahead of it into the pre-recruitment region. And again, this is happening all the way around the microelectrode, or all the way around the ictal wavefront. And what you'll notice here is if you look at the directions of these fast traveling waves, they change. So initially, these fast traveling waves are heading in parallel to the direction of the ictal wavefront expansion, and then they're heading in the opposite direction. And this is the key insight of this ictal wavefront theory, is that these traveling waves reverse direction after the ictal wavefront moves across the microelectrode array. Okay, this is a cartoon. Let's see an example of this in a human subject. So here is an example. What's plotted in this figure is first the ictal wavefront direction is indicated by this black arrow. These other arrows indicate the inferred directions of these fast traveling waves during the seizure. And what you'll notice is that there are light colored arrows and dark colored arrows. These light colored arrows indicate times, pre-recruitment times, before the ictal wavefront has moved across the microelectrode array. And what you'll see is that these light colored arrows, they tend to align with the direction of the ictal wavefront. So they're heading in about the same direction. So these fast traveling waves are being sent off ahead of the ictal wavefront. The ictal wavefront then moves across the microelectrode array. We're in the post-recruitment region. And after the ictal wavefront passes over the microelectrode array, these fast traveling waves are going in the opposite direction. So they're moving in the opposite direction, approximately from this black arrow. It's pretty convincing evidence that this is seen in vivo data in humans. Okay. So ictal wavefront theory, there's a sudden change in traveling wave direction, these fast traveling waves, after the ictal wavefront passes over. So the alternative theory, which I'll call the New York hypothesis, is that an ictal wavefront emerges during a seizure and it drives traveling waves in two opposing directions. So you might conceptually think of this as we have this ictal wavefront that's sending waves into the interior, into the exterior, and as soon as this ictal wavefront crosses our microelectrode array, we get this shift in the fast traveling wave direction. Okay, so also I'll note in this study, there were 3 patients. Again, really hard to collect these data. So both the New York hypothesis and Boston hypothesis based on observations from 3 different patients. So there are at least 2 competing theories for the origin of these fast traveling waves. There's certainly agreement that these traveling waves exist during human seizures. I showed you a movie, right? It didn't require any data analysis. We could see these fast traveling waves moving over the brain during a seizure. So we all agree on that. The disagreement is what are the mechanisms that could be producing these fast traveling waves? There's the New York hypothesis that these fast traveling waves come from an ictal wavefront. And then there's the Boston hypothesis is that they emerge from this fixed cortical source that we could see, especially late in the seizure. Okay. how can we resolve this? Which one is right? And really, what's the source of this conflict? Both groups looked at a small number of subjects. We applied very different data analysis methods to infer the direction of these traveling waves. And maybe it's this variability we're seeing in subjects or in our analysis methods that's leading to these different theories. So to help resolve this conflict, a very talented graduate student in our group, Emily Schlafly, she was able to aggregate the data working with Kathy Chavone and Ed Merricks at Columbia University, Sid Cash and MGH, aggregate the data from New York and Boston, which at that point consisted of data from 11 patients and 31 seizures. So take all the data from both institutions and then apply a consistent analysis pipeline where we'll have a consistent way of detecting traveling wave events, estimating their direction, and then identifying intervals of stable traveling wave direction across all the patients from both of the institutions. And I'm going to tell you about 3 of the results from Emily's paper and help to resolve the disagreement between these 2 hypotheses. Okay, so the first observation is that these ictal wavefronts, this increased firing, tonic firing of the multi-unit activity, occurs in most seizures. So here's an example of a ictal wavefront. What's plotted here is a microelectrode array, a 10 by 10 grid, where the time of arrival, TOA, of the ictal wavefront is indicated. Light indicates the ictal wavefront arrives early, and then black indicates it arrives later. And so what you see here is the light colors are at the top left, the dark colors at the bottom right, and this arrow is indicating the approximate direction that this ictal wavefront is moving across the microelectrode array. This is 1 seizure, 1 patient. 3 seizures from this patient give consistent results, this ictal wavefront propagation. And what Emily also found is that the features of these ictal wavefronts are consistent with what was reported previously. So there's some firing rate near 100 hertz. This ictal wavefront lasts for about 2 seconds at each microelectrode, and it has some speed about 1 millimeter per second. So looking across all the patients from these 2 populations at MGH and at Columbia, we get consistent results with what was reported earlier. So these ictal wavefronts exist. The second result that Emily found is that the ictal wavefront has a transient impact on the traveling wave directions that are consistent with the New York hypothesis. And let's look at an example. So here's one example recording from 1 patient and 1 seizure. This is a busy plot, so let's look at the different parts. So what's plotted on the vertical axis is the ictal wavefront direction. This is the direction of the fast traveling waves shown here. And everything has been rotated, so the ictal wavefront direction is at 0 degrees. And here we're inferring the direction of these fast traveling waves using 2 methods. One method is consistent with the Boston hypothesis that's shown in light blue, and the other method is consistent with the New York hypothesis that's shown in purple. They give approximately the same results. So you could look at either one. And if we look at this plot, at time 0, the ictal wavefront is going to move across the microelectrode array. So what is this plot showing us is if we look before the ictal wavefront arrives, so at negative times, and look at the direction of these fast traveling waves, they tend to be near 0 degrees. So they tend to be roughly aligned with the direction of the ictal wavefront. They're heading in about the same direction as the ictal wavefront. Well, after ictal wavefront passes, there's a shift, and now these waves are approximately reversed, maybe 90 degrees, maybe 180 degrees. These waves are going in a different direction after ictal wavefront passage. And that's consistent with the New York hypothesis. There's a ictal wavefront, it's driving waves in 2 directions. After it passes, those directions change. So this example is consistent with that. Again, this is one example, 1 seizure. Let's look at the population results for all patients and seizures. And that's what's shown here. What's on the vertical axis? The absolute fast traveling wave direction relative to the ictal wavefront. So 0 degrees indicates those fast waves are aligned with the direction of the ictal wavefront. 180 indicates they're going in the opposite direction. The fast traveling waves are moving opposite of the slow movement of the ictal wavefront. And then again, we're estimating with 2 different methods. They give consistent results, and the ictal wavefront passage is at time 0. And if we look at this plot, what are those fast traveling waves doing? Initially, they tend to be aligned with the ictal wavefront. Their absolute traveling wave direction tends to be near 0, or at least less than 90 degrees. After the ictal wavefront passes, that direction changes, and they tend to be roughly reversed, heading in the opposite direction of the ictal wavefront, at least greater than 90 degrees. And then it becomes more complicated, what happens later in the seizure. So this observation is consistent with the New York hypothesis that the ictal wavefront has an impact on the direction of these fast traveling waves. And here we're showing that it's at least in the observations we're seeing here, it's a transient impact. Okay, so that's the second result. The third result that Emily found is that multiple shifts occur in the traveling wave directions during seizures. So here is an example from one subject where, again, we're estimating the traveling wave direction. This is using the Boston method. Each black dot indicates the direction of a fast traveling wave. And again, 0 indicates the ictal wavefront direction. So that's the direction the slowly moving ictal wavefront is going. And if you look at this plot, what we see here is that there are 3 stable intervals of traveling wave directions indicated by different color. By stable, I mean the direction of these waves is not changing too much. Maybe you could see that by eye. We have directions that are kind of scattered in here, and in here at different times. Again, ictal wavefront passes is 0. What I like about this example is we see aspects of both hypotheses in this single seizure. So if we look around the ictal wavefront passage, this first part looks like a shift at the ictal wavefront, which is consistent with the New York hypothesis. Initially, these fast traveling waves are heading in the same direction as the ictal wavefront near 0. The ictal wavefront passes, and then they're going in about the opposite direction. But if we look later in the seizure, there's some other direction that emerges, and this looks like the Boston hypothesis, a fixed source of these traveling waves. So looking across the population of patients in seizures, Emily found that in most of the seizures, there are 3 or more of these stable traveling wave directions, and one of them tends to dominate. One of them tends to be the longest, and that one tends to occur late in the seizure, consistent with the Boston hypothesis. Okay, so the results summary. So consistent with the New York hypothesis, we did see ictal wavefronts occur in most seizures across this population of patients. The ictal wavefront has a transient impact on the traveling wave direction. And then consistent with the Boston hypothesis, there are multiple sources of these traveling waves and one that dominates, especially approaching seizure termination. Okay. So with those observations, can we go back to our model and think about what mechanisms could produce these in vivo traveling waves? We're going to consider the same mean field model, but we're going to include 2 sources of traveling waves. One source that's consistent with the New York hypothesis and another source that's consistent with the Boston hypothesis. So what does that mean going back to our cartoons? In our model, we'll have an ictal wavefront. It'll be sending out waves in 2 directions ahead of it and behind it. So in this blue direction and in this red direction, and the ictal wavefront will slowly expand over our simulated surface. And we'll include a source consistent with the Boston hypothesis, this radio tower that's going to be trying to send out traveling waves throughout the seizure. Okay, so before jumping into that new mean field model, let's again look at a patient example. So here I'm going to show this is an example from a single patient in seizure. What we're indicating here is, again, estimated traveling wave direction of these black dots. These stable intervals correspond to intervals of time where that direction is relatively stable. And again, 0 indicates the direction of the ictal wavefront, the slowly moving ictal wavefront. So what happens? Early in the seizure, there's an ictal wavefront. We're imagining there's this ictal wavefront that emerges. It's sending out waves ahead of it, right, corresponding to these waves that are aligned with the ictal wavefront. The ictal wavefront slowly passes. It reaches kind of the exterior of our observation window. And now it's sending waves back into the interior. So we have these waves that are these fast traveling waves that are moving reversed relative to the ictal wavefront. And then sometime later, something else emerges. The ictal wavefront is maybe dissipated or it's too far away. We could just see this fixed source and we see these fast traveling waves in another direction. So again, this is a cartoon. This is real data on the top. And I'm trying to suggest that this cartoon is at least qualitatively consistent with the types of things that we see in the in vivo data. Let's look at a simulation of this model where our goal is to capture these 3 stable intervals. We have wave directions that correspond to the ictal wavefront aligned and reversed, and then another fixed source direction that appears later in the seizure. So here's a brief description of a simulation result, where here again, we'll simulate a cortical surface. We'll have an origin of the ictal wavefront. The ictal wavefront will start at this star and then slowly move out. We'll have our fixed source, this radio tower that's always trying to send out fast traveling waves. We'll have our microelectrode array. So imagine we're recording right here. And then the gray region is going to indicate what we'll call an irritative zone. This is a region that can support fast traveling wave dynamics. Slightly more excitable than the rest of the surface. Okay, so let's look at a simulation of this model. we're plotting here, again, a bunch of different things. This gray line corresponds to the activity of one node in our network. So this is the excitatory population activity. And then we're indicating the direction of the fast traveling waves. These correspond to these black dots relative to the slowly moving ictal wavefront. What's happening here in this simulation? Initially, the ictal wavefront emerges. That corresponds to right here. This ictal wavefront emerges. It's sending out waves ahead of it into the pre-recruitment region, and that's passing across the microelectrode array in a direction aligned with the ictal wavefront movement, so near 0 degrees. As time evolves, the ictal wavefront is slowly expanding outwards. It eventually crosses over the microelectrode array. These black dots correspond to the ictal wavefront, and it reaches the other side of the microelectrode array. It's now sending waves back into the post-recruitment region in this red direction, and that corresponds to these waves that are reversed relative to the ictal wavefront. Eventually, the ictal wavefront, it hits the boundary of this irritative zone, it dissipates, and all that's left is our fixed source, and that fixed source is now sending out waves that our microelectrode can see, corresponding to a different direction, shown here about 60 degrees. So this picture that I'm showing here is at least qualitatively consistent with the picture I'm showing above. We can shrink these timescales depending on how quickly we make the ictal wavefront move. We can place this grid in different locations and you can get different types of dynamics. And again, I'll just mention that this model is able to produce a lot of the features that we see in vivo. So a lot of the in vivo observations. So what is the resolution of this story, these two competing hypotheses? Well, hopefully I was able to convince you that there are multiple sources that can produce these fast traveling waves during seizure. The ictal wavefront is consistent with the New York hypothesis. We see that in the data. We do see evidence of this fixed source, especially late in the seizure, that's consistent with the Boston hypothesis. So a very diplomatic answer. Components of both theories are correct. And that's where I'll leave that there. And then I'd like to thank the organizers for inviting me and accommodating my schedule, and also the patients and the hospital staff who make all this data possible, all of our collaborators and our funding sources. And I'm happy to chat more. I think that was 40 minutes. Happy to chat more if you have any questions.
Thanks, Mark. This was awesome. I think there'll probably be a number of questions. So everyone, you can start.
I have a bunch of them.
Yeah, Bard, do you want to get us started, and everyone else can
Sure. Okay.
in the chat.
So I guess the Boston hypothesis is there is a source that stays on the whole time, and then you turn it off.
Yeah, it may stay on the whole time, or it may be ignited by the seizure activity at some point late in the seizure. But yeah,
But in your model, I'm talking about in your model, it stays on all the time.
Yep.
So I guess the question is what turns it off?
Yeah, good question.
And the other question, I have a couple other questions. In the what's the mechanism for that spatiotemporal chaos you see preceding the spiral waves?
Yeah, let me pull up the picture again. That's all right. I'll share my screen again.
And I have one more after that.
Yeah, okay. Okay, let me go back here. Can you see my slides?
No,
Not anymore? Just me. One second. I'm going to go back to the picture I think you're talking about. So
it's one of the first ones you showed of the model.
Yeah, I got it here.
Where you have all that weird little
yep.
like stuff.
All right. Now you should see a big red screen.
Yep.
Okay, 1 second. Now you see.
There we go. That's the picture. Yes.
There.
Yeah.
Okay. Yep. So you're asking a couple of things. I think the second question was what's causing all this stuff in the middle?
The little black dots and stuff. The first 1, 2, 3, 4 frames 5 through 7 or 8.
Yeah. Okay. Good question. So my quick answer, I can tell you what I understand about it, and then maybe you'll have more insights. We initially ignite this source right here. So we ignite this source. It sends out a wave that rapidly propagates over the whole surface. You can't really see it here, but you can see the first manifestation of it.
Yeah.
This boundary goes quickly over the whole surface. And I think what that's doing is it's exciting some populations. So there's also noise in the system. It's exciting some of the populations, and then those happen to stay on. Other regions are inhibited because those excitatory populations turn on inhibitory populations. So it's possible they could knock down some other regions. And then that stays on for a while. And then as potassium diffuses more and more, the surface becomes more excitable. But what's actually producing these patterns, I don't know.
The
You
lower
tell me.
stuff is almost solely due to the potassium dynamics, I guess, then, right?
That would be the slowly evolving parameter that's increasing the excitability. And it's almost consistent over the surface. I think a fair approximation is you're increasing uniformly the excitability of the whole surface.
And then my last question is, do you impose the ictal wavefront as a sort of a slowly moving excitation that just propagates across?
Right. Yes. So
Oh, okay. All right. That's how you do it. Okay.
it makes a random jump at each moment in time, but it's kind of moving slowly outward. So we impose all that. And I think you also asked what causes the seizure to stop. We do it by hand in the model. I don't know. I don't know in vivo.
Yeah. All right. I'll let other people ask questions.
Okay. We got a few in the chat now. So I will actually just go with one of the earlier ones. There's a question from Srinivas Kota. I'll put you up on stage to ask. You should be able to unmute or start your video if you'd like.
Hi, thank you for a great presentation. I was wondering, can we see traveling waves in scalp EEG?
So probably, yes. The difficulty, I think there's the spatial resolution will be lower. Maybe if you had a high density EEG recording, maybe you could do it. I'd be worried a lot about artifacts in that type of recording. But maybe if you had something like a slow oscillation during sleep, maybe. And I'm not sure during a seizure. So I don't know the answer during seizure on the scalp EEG.
Okay, and is it traveling waves? Can we predict seizure using traveling waves?
So again, I don't know the answer. One thing we've thought about is if you can find where these traveling waves are coming from. So if you can localize the direction of that, would that be a target for intervention? We've started thinking along that path, but we have not completed that story. So I think because these traveling waves tend to emerge late in the seizure, they wouldn't be useful for intervention at that time because the seizure's already started. Instead, they might be useful for targeting where their origin is.
Okay, thank you.
Mm-hmm. Good question.
We have a couple of hands raised. There's one from Henry Asher. I'm gonna let you unmute and get on stage as well.
Nice talk, thank you. My second question follows up on the one that was just asked, so you don't need to, and it's, have you tried to link this fixed source and the ictal source to surgery outcomes? But the other thing I was wondering about is how consistent this sort of separation distance between the fixed source and the ictal source is.
Yeah.
And you also mentioned that sometimes, I think you said sometimes you see multiple fixed sources. So I'm wondering if there's any patterning to that relationship as well.
Yeah. Okay, good. Yeah. So the first question, can you use this to target? We've thought about that. The N is so low, at least for the microelectrode arrays. We don't have an answer yet. So I don't know the answer to that, but we've thought about trying to do that. And then your second question was like, how much does it matter where we put these things? The ictal wavefront and the fixed source, I think was your question. It matters a lot. And if you look in Emily's paper, you'll see different scenarios where if you put your grid here or here or here, you get very different types of wave dynamics that can give you different numbers of stable patterns. So it does matter. I just showed you one example that was consistent with the data we see, but it matters a lot.
Thank you.
Did I get all of them?
Yeah, I think you got it. Thanks.
Okay.
I have kind of a related question. What happens when the ictal wavefront hits the basically passes the fixed source?
Yeah, so in this model, they would just pass over each other in the way we've simulated it.
Okay.
The waves are they're destructive, so they don't pass through each other. If you have fast traveling waves hit each other, they collide. So nothing would happen in our model, and I don't think enough is known about the in vivo data to say what would happen in vivo. You'd have to get very lucky to have a microelectrode array that could see both.
Yeah.
Because they're so tiny. Yeah, I think that would be very, very rare. At least we haven't seen it.
Okay. All right, next I'm going to put Sophie Ack up.
Hello,
Be
can
able
you
to-
hear us?
Yeah.
Mark, thank you for a really interesting talk. I was wondering about the pattern of from the fixed source. After you get the wave from passing, it seems to settle on a fixed direction. How much of that is there anyway when you're not looking at seizures, when you're just looking at the normal function of the brain? Is that just a return to the normal functioning, or could it be competition between different sources that actually is dampened by the seizure, that now this pattern is dominating, if that makes sense?
Yeah, good question. So we don't have observations, at least in this study, we don't have observations in outside of the seizure interval. We can look a little bit before the seizure, which is maybe indicative. It's probably not the normal brain or normal brain functioning, but we do see some evidence of traveling waves, and they're all over the place. So here's one example where you look at where are these fast traveling waves coming from before seizure onset, lots of different directions. So I can't say what's happening during normal functioning. At least in the old days when we were collecting these data, they were very heavy, right? So trying to store all this data during a 24-hour chunk and trying to get cognitive time would be difficult, at least the way we were doing it back then. So I don't know.
So you're saying it's just the normal traveling waves are just not stable. There's not a fixed direction that they happen in.
That's not what we see, and it also, at least, it doesn't feel consistent with these seizures, which are these really pathological synchronous events that are moving across the whole brain. There might be other scenarios where the brain would want to do something like that, but it's probably limited to particular states.
Okay, thank you.
Yeah, good question.
Okay, so next, Ganendra Singh, would you like to ask your question?
Yeah, hi Mark, really interesting talk. Initially, you showed with MIA recordings that there were quite different kind of amplitudes were passing through. So I was interested that do you observe different characteristic shapes of multiple amplitudes? When you showed that video in the starting, that I observed there were three, sometimes there were two. So I was curious that do you observe different such characteristics?
And so you're wondering, like, you're looking at these
Yeah,
shapes
here,
maybe over here?
exactly.
Yeah,
Yeah.
so we tried to think about that a bit. And this first front is very regular, tends to be very regular. You see this nice traveling wave. Afterward, what happens next? It seems to be spatially organized, but it's very complicated to figure out what's happening there. So I agree that there's this initial front that passes across, and then something else happens. We don't know what that is. Our thought is that it's locally generated. So you might imagine you have this wave of excitability that sweeps over the surface, and you're activating some local circuits that do something, right? That almost looked like a wave that emerged from the center, looking at the microelectrode array. So you have this wave of excitability that propagates over, that's being exposed externally, and then something internal is happening in this region. But that's as far as we've gotten.
Yeah, this is interesting because especially during, let's say, spirals, these amplitudes quite changes a lot. So I was wondering whether it's interesting to really characterize them. Again, thank
Yeah,
you for your
you can see it here. Yeah, exactly right. So we have these lower amplitude things that follow these fronts. We've thought about this a little bit. If you really want to dig into these details, you could look at Emily Schlafly's thesis, which has it at the very end. But it's a very hard problem. One reason we found it so hard is that the things that happen afterwards are so diverse. It's not like these fronts, which are relatively easy to characterize. What happens afterwards is all over the place.
Yeah, I agree completely. Thank you so much.
Yeah, good question.
Next, Sangan, would you like to ask your question?
Hi, thank you for the wonderful talk. I was just wondering if you looked into, say, if there's more than one ictal wavefront origins or more than one kind of like cortical origins, what happens then?
Good question. Yeah, we did not look into that. I don't have an answer.
Thank you.
Yeah,
Okay.
yeah, good question.
All right. Next, Karbai Zhang, would you like to come up and ask your question?
Yeah. Thanks for great talk. I'm just wondering, like, can you see such spatial temporal dynamics during the interictal spikes?
Yeah, I would like to know the answer to that. We don't have the data for it. So the data that we collected were only around the seizure. So I don't know the answer from the data that we've looked at.
Okay, thank you.
Yeah, probably. Actually, I don't know. I don't know.
Looks like Bard has another question.
Okay.
Bard,
So,
do you want to go ahead?
so in your combined model with the wavefront going off and the emitting waves and things like that, I gather the emitting waves from the source occurs in the Boston model occurs only long after all that spatiotemporal chaos and stuff goes away. So what happened to that when you initiated this simulation? Shouldn't there have been that spatiotemporal chaos ahead of the place where you fix the source, like you
Yeah.
see in the first thing, and then that ictal wavefront doesn't seem to generate? Why doesn't that generate that spatiotemporal? I imagine you're just activating it in a spatially moving way.
I think the main reason for that is we've already increased the excitability of this irritative zone. So it's already prone to propagate waves.
Okay. Got
And
it.
because the ictal wavefront is closer to the microelectrode array in this simulation, it's the one that's dominating the waves. This radio tower, the fixed source, also sending out waves, but it never makes it to the microelectrode array because it's being knocked out by the waves coming out of the
Right.
ictal.
Right.
And you can imagine if I were to move this microelectrode array right next to the fixed source, that's what we would see.
You'd start to see the other
Maybe.
stuff. Okay.
Yeah. And if we turned off this irritative zone, you would. I expect you're right. You would get that kind of spotting pattern first, and the little spirals or crescent moons, those locally traveling waves.
All right. Thanks.
Good questions.
Great. There's just a couple more in the chat. I know we're kind of running up on time. So Mark, are you okay with
Yeah, I'm
the
happy to answer. Yeah,
Okay.
if I can.
Omer Sharon, would you like to come up and ask? I don't know if he's still there. Oh, okay. I don't think he's still there, but I can read his question.
Sure.
What's the relevance to the healthy brain? Do you think we can learn something on how traveling waves work in general, or do you think we're seeing them in an exclusively pathological mechanism?
I think we've chosen a system where it's easier to see these waves. So I think that's part of it. My hunch is that if we were to look in the healthy brain, we would not see waves that are propagating over the entire cortical surface unless it's in some scenario where the brain would want to do that, maybe during sleep. My hunch is that if you were to look in the healthy brain, you would see more things like these reverberations that happen afterwards. We have this wave that propagates over. I guess you'd see more locally generated activity, but I don't know the answer.
Yeah.
We're in this pathological state where you get these waves that are driven across almost the entire cortical surface, and then we happen to see them on the microelectrode array. So it's simpler than I think what that question was asking.
Yeah. Okay. We have another one from Michael Bick. Looks like you're still here. Would you like to come and ask?
Sure, yeah. Thank you for the great talk. I feel like my question was kind of answered in the discussion, but I was thinking about sort of like the nature of the origin of the seizure. Like you have this ictal wavefront and you have like the radio tower origin. And sometimes you see different stable numbers of stable points because of that. Do you have like any insights as to like any relationship between either like the distance between them or like what causes that or anything in that nature?
Yeah, it's a great question. I don't know the answer. You might imagine that if you constrain the system enough, like if you constrain the system and say, I believe there's an ictal wavefront, I believe there's a fixed source, and I have this in vivo data, maybe you would be able to use that to try to estimate where those things are because you never get to see them directly, especially on the microelectrode array. So maybe if you constrain the system enough, you could try to make estimates of where those things are. But I think that model would be dominated by the constraints you impose. And maybe if you had a bunch of microelectrode arrays and you can cover a brain in them, you'd be able to answer that, but that's not possible.
Right. Yeah, that makes sense. Thank you.
Yeah, sure.
I have one quick question. I keep it short, but great talk. I'm curious about the mean field model. Do you have any intrinsic noise in it that can make these ictal waves happen by itself? Did you ever try it or?
Yeah, so I could tell you a little bit about. So the noise that we have in the model is we allow the excitatory population activity. We just have a stochastic noise term that we use to drive it. That's the only noise that we have, at least in the initial setup of the model. And then what was the second part of your question?
Yeah, I'm curious if it's feasible to set up a noise level that make the whole system autonomous and generate those kind of waves itself.
Yeah, it's an interesting question. We have not gotten to that point. It would be nice if you could get it to kind of induce its own focus, right?
Yeah.
That area would probably have to be predisposed in some way, like slightly more excitable or maybe more connected in some way. We chose the simpler path of just trying to characterize these waves when we control that.
Yeah, I'm just saying because the hypothesis we don't talk about is we think the source of these noises are the stimulation you do in the model, and we don't know what makes it happen in the brain. So
Right.
something is probably there's a charge coming to a specific source.
Right. I just turned this node on, right? It would be nice if that
Yeah.
was somehow part of the dynamics.
Yeah.
I agree. I agree.
Great.
Very cool. It looks like we have one last question in the chat. Pega, would you like to come and ask?
Yeah, thank you for the great talk. And my question was kind of, again, comparing these waves, especially in your model, with the more physiological oscillations. For example, something like beta, it's pretty amenable to modulation with movement or any sort of like kind of action or physiological changes. But do you use any sort of perturbance? I'm not really familiar with modeling work, but like any sort of factor to test these waves, because me coming from a medical background would be, my hypothesis would be these are kind of like stubborn waves. You probably can't stop them easily. How can we test for that?
It would be interesting if, I guess, if you had a subject that had some kind of neurostimulation, you might be able to try this cortical, I guess, be RNS, right? Potentially. It would be interesting if you could stimulate somewhere along the path of these waves as they propagated over. So that'd be a challenging problem because you'd have to detect them in real time and try to stimulate. That might give you some insight into the underlying mechanisms. I don't think it would help. I think you're right. It'd be stubborn. My guess would be the waves would just propagate around it, and it probably wouldn't help treat the seizure because at this point, stimulation far away from that source, the waves are already going, right? So that's not going to help. It might slightly disrupt the one wave that happens to go over that region, but I think the waves would continue on, like you said, stubborn.
Thank you.
Very cool. Okay. Well, if there aren't any other questions, I think we'll wrap up. This was awesome, Mark. Thank you so much.
Thank you. Yeah, thank you for the invitation. It was a lot of fun.
All right. We have a bunch of really great speakers coming up the next couple months, so stay tuned. And, obviously, if you miss any talks, catch them on YouTube as well. All right.
Thank you.
Thank you.
Take care.
All right, that was great.
Good to see you, Mark.